Hey, biophysicist. Welcome back to a special edition of the podcast. So we have today a bit of a new kind of format that we're trying, and also some new hosts to kind of introduce. So with us here, we have Avaneesh and Maxime. They're two researchers that are going to be joining in to help host the podcast as well. So I'm kind of excited to see what they have to show. So Avanesh, if you maybe want to take a second or two and introduce yourself.
Hi, everybody, and thanks, Clark. My name is Avneesh. I am a PhD candidate at the University of California, San Diego, doing mostly theoretical stuff on coexistence and population behavior in microbial systems. Um, yeah, I don't know if there's anything else you'd like me to say.
No, that's perfect. So Maxime.
Yeah, so my name is Maxime and I'm a PI in biophysics. I'm studying bacteria from an experimentalist point of view and also somehow theorist point of view. And I'm based in Paris at the ESPCI, where I got a lab called Laboratoire Biophysique and Evolution.
Cool beans. So today we're, kind of introducing the new hosts, and we thought it would be fun for each of the three of us to pick kind of an influential paper in your field and discuss it. So the idea is basically one of us is going to talk about a paper, and the other two are going to kind of squeeze in a couple questions. And in that regard, we're just kind of chat about some biophysics that we think is pretty influential. So, Avanish, I think we'll start with you again. So, if you want to introduce your paper and tell us a little bit about it.
Perfect. Thank you. So the paper that I'm going to be talking about today is called Coexistence in a Variable Environment, and it was written by Richard Levins, who's a population biologist, a very famous one. And the paper came out in 1979 in the American Naturalist, which is primarily an ecological journal, but has far-reaching influences in biology. And this paper, it's not as well known as other papers in my general area of interest, but I think it really um, addresses a very important question at the heart of community ecology and species diversity. So there is this general question as to how diversity arises, and if you look at if you write down a simple set of equations and then you ask, well, I want to study the system when things are no longer changing, so when things have reached a sort of equilibrium, then you find that the number of species that exist cannot be more than the number of resources that there are. And this was established by a lot of different people. um, Chief of them were Lotka, Gauss, Volterra, who worked on this in the 1920s. And the result that comes out of it is known as the competitive exclusion principle. And they used a mathematical model to demonstrate that you can only have as many species as there are resources. Uh, does that make sense to Clark and Maxime? Is there any question based on this?
Yeah. So, when you say resource, what exactly is it like? Available food, available location around the cell. Like, what do you mean in terms of a resource here?
That's a great question. And honestly, it's left a bit vague. Um, Mathematically, it is whatever allows a certain species to grow. Um, Biologically, that resource could be an abiotic resource like a carbon supply or a certain other mineral, or could even be light. But abiotically, it could also be a biotic resource, like, for example, heterotrophs eating phototrophs. So uh, grass for cows would be a resource. And both of these are addressed by the same mathematical principle. If you want a single resource to be give um, support a certain number of species, then you can only have one species. not because that single species will be very, very good at consuming that one resource. If there's another species that is either worse or better, if it is worse, it'll lose. If it is better, then it will win. And so, almost tautologically, if you're only looking at equilibrium solutions where nothing is changing, then you can only have one species survive on one resource.
Is there any type of, competition or maybe like a hierarchy of things, right? So having one species eat another that eats another, or is this all like kind of independent, no intermixing or no convolution between the different species and the different resources?
Right. This is a great question, and a lot of people have tried to address questions like this afterwards. On one level, if you were to just imagine a series of levels which are disconnected from each other, so trophic levels, as we are taught in food chains in elementary school, right? That is a fairly easy question to answer. And basically, what it turns out is the number of members in one trophic level has to be less than or equal to the number of species in the trophic level below it. So if there are 5 plants, then it can only be 5 plant eaters, and there can only be 5 or less um, things that eat the plant eaters and so forth, right? And that is why you see that the number of species that are at or at least this is believed to be why there are very few predators and very few apex predators. There is also an energy balance that is involved in it, so it's a lot more complicated than just that. But this is one ecological principle that tells you that you can only have fewer consumers than producers.
Yeah, I think it's also a great question because at the beginning of the article, the Levins point out that there is a weird dichotomy that we do between organism and environment,
Hmm.
and that actually they are not that much separated from each other. Like organisms are shaping the environment, and sometimes environment are organisms. Like the example you just gave, if you are a grass eater, you eat an organism, and the grass is an organism, but it's a resource in the end. So I think the best well, what I grasped from the paper is that the resource is something that can be directly consumed by an organism or is produced by it. It's just the input and the output, and it can be whatever an abiotic thing or a biotic thing.
Right. No, absolutely. And so, we earlier learn about the food chain, but then once you go to high school, they tell you, oh, no, no, it's more like a food web, right? Because you have different things consuming different things and producing them. And then it becomes a little bit more complicated. And then what is a producer and what is a consumer becomes a bit more difficult to differentiate.
Mm.
And there is a lot of theory around that as well, but now depends a lot more on the structure of the interactions that you have built in and the structure of the entire network of different species that are at play.
Mm. And in this sense, the paper is beautiful to keep this generality and explain what you are going to explain to us, I guess, in the
Yeah.
following.
No, absolutely. And so there is, because you have this notion that you can only have one species surviving on one nutrient, or 5 species on 5 nutrients. Uh, it, it then a concept that came about was the concept of an ecological niche. And this has become very popular in ecology, where basically you say, okay, this species is a grass eater, so its niche is eating grass, right? And then over time, this notion of a niche became more developed, but also more vague in a sense, and to the point where the definition of a niche right now, if you ask many people, is kind of circular, where they say a niche is something that only one species can survive on, whereas the entire principle was established because there was this mathematical observation that a species can only survive on one niche. So it's a bit of a tautological definition that exists, and it makes quite a few people uncomfortable to be talking about niches because it can really come to mean anything. But it
I
is,
guess
yeah.
in some sense, it's better to say only one species can thrive in this particular niche, because I'm imagining that you're assuming that none of these species are really kind of evolving with the times, right? Like you can never really adapt to whatever circumstance you're presented in. Or is this two different time scales? I guess the time scale of kind of natural evolution versus like being able to reproduce well in a particular ecological environment.
Right. No, absolutely. There is a timescale separation involved. The ecological timescale is generally taken to be shorter than the evolutionary timescale, though people are pushing back on that. But what you're addressing actually is kind of gets at the heart of this result, which is survival of the fittest, right? Darwin's hypothesis, and the entire theory is based on saying that you have this um, Malthusian population pressure, such that it's not just that the fittest thrive, it's that only the fittest survive. It's not that you can be slightly less fit than someone else and like eke out an existence. No, you have to be the fittest to survive. Um, and this survival of the fittest is really at the heart of both ecology and evolution, because if you want another species to gain a foothold, then it has to demonstrate that it is um, fitter than the existing species. And that's how natural selection has allowed for such rapid progress to take place, um, that we evolved from bacteria. Okay, perfect. So moving forward, this was a bit of a conundrum because, like putting the competitive exclusion principle, because we see so many different species. Like if you were to go out into the soil and just scoop out like a cup, and then if you count the number of microbes living on it, there's thousands and thousands of microbes, right? Um, and there are not that many resources in the soil. So what gives? How are there so many species that are coexisting on very few nutrients? And this has been a very fundamental question in ecology. Um, Famously, there was this paper called "The Paradox of the Plankton" that Hutchinson wrote, where he posits this question and he answers it actually by saying that fundamentally, when Lotka, Volterra, and Gauss, when they were coming up with their theory of competitive exclusion principle, they supposed that everything was at equilibrium, whereas there is no reason to believe that everything would be at equilibrium in the actual environment, because things are constantly changing around us. And these variations can come both because of external changes such as seasons and day night cycles, and so forth, but they can also come because of internal changes that happen, because as Maxime was saying, the species shape their environments. So if they shape their environments at different rates, you can end up with a scenario where you have a constantly changing environment. And this is what Richard Levins is trying to address in this paper, and trying to come up with a theory of how you can address species diversity in a constantly varying environment. And he has a lot of other grievances as well that he is trying to address here, exactly as Maxime said, that typically you have this separation of the roles of consumer and producer, which was partly actually his own doing in a paper that he had written a decade ago, where he had outlined consumers and resources, and come up with this new formalism that has become very popular. But he felt that that went a bit too far. So he wanted to discuss how you can work with a constantly varying environment. And what he provides is a very simple recipe where you take a time averaging, but this time averaging has to be done in a very careful manner. And when you do the time averaging, then what you notice is that if the uh, dependence of the consumer on the resource for its growth, if everything is linear in the system, then there is no difference between the time averaged result um, and the constantly varying result, such that you're only going to end up with with the competitive exclusion principle. The number of species is less than the number of the resources. But if you have any non-linearities in the system, which is typical, like in any biological system, linearity is kind of an absurd thing to hope for, because when the entire system is saturated with things, then it behaves very differently than when it is it has very few of the things. Biology does not respond the same regardless of the amount of stuff present. It adapts in some way. And in that case, then the nonlinearity, as he showed, it can actually be treated as a resource in itself. And that's because when you take the statistics, then when you time average everything, then you get the you get that the mean of the entire variation is the mean of the entire resource is sorry, what the effective time averaging is the mean plus a variance term or higher order moments as well that could come in. And these higher order moments can effectively be used as a resource. And I won't get into the math of it. It's tough to do that in a podcast. But what the beauty of the paper for me was showing that you can effectively create resources out of nonlinearities in the system. And in a way, you actually get that the species consume these nonlinearities, and the result that he's ultimately able to arrive at is that the number of species is limited by the number of resources plus the number of nonlinearities, because the nonlinearities effectively act as a nutrient. um,
Is
yeah.
there some type of property about the nonlinearities that has to be satisfied? Like, you can't have an infinite variance if you're integrated across some span, or is there some, like, do we have to have nice nonlinearities, or can it be any nonlinearity that we want?
No, really, yeah, it turns out you don't need any nice nonlinearities. And essentially, the reason why this is because if the nonlinearity is encoded in the function f of x, then the expectation of f of x is not equal to f of the expectation of x when you have a nonlinearity. And so when you do this, you end up getting another term, which is the variance or the covariance or higher order moments. And these other terms can effectively be used to balance the equations. So one of the reasons why you end up getting that if you have n resources, you can only have n species, is because you can only have n solutions to this complex linear uh, calculation. But then when you go to this nonlinear setting and you make this time averaging, then you effectively end up getting these other terms that can also be used in this system of equations in order to satisfy the survival of multiple species.
Yeah, I really like this idea of feeding organism with non-linearities. I think it's fascinating to think in terms of this balance equation. Honestly, I was really struck by this demonstration. I mean, I realized something there. Mm.
Yeah, I mean, I guess it's kind of, your mother saying, be sure to eat all your nonlinearities if you want to grow big and strong. So,
Right. Takes me back to the days when my mom used to say that. Yeah, what I completely agree with you, Maxime. Like the technique that he provides is just so simple and so beautiful and produces such counterintuitive results. I think the interpretation of it is what is really beautiful in this paper. Um, for me, like as someone who does a lot of statistical physics, it really drives home the point that coarse graining that we typically do in statistical physics cannot be done just simply ad hoc. You know, if you are trying to coarse grain a system, then you have to make sure you're accounting for all of the different effects, different interactions within the system. And if you're able to do it mathematically, then you can have a coarse graining which has slightly different predictions. And in some cases, these different predictions can lead to very significant consequences. Um, and in this case, it really is laying the foundation for non-equilibrium ecology as a way to explain diversity in the system. Because you're saying that if you have temporal variation, which is granted, and when you have nonlinear dynamics, which is also granted, then you have an emergent phenomenon which you do not get from either of the two, and which you would not expect just from thinking about what either of the two are doing to the system. It's the interplay of the two that, when you take the statistics of it, turns out give very counterintuitive and very profound outcomes as a result.
Mm. We could use this framework to ask the question, but in reverse. If we know all the species on Earth, could we infer all the nonlinearities that there is in the interaction between them?
That's a great question, Maxime. Um, Solving inverse problems is very difficult. I hope you're not volunteering me to do it, but I agree. It would be very impressive, because what we do have as an empirical observable is that there are a lot of species.
Yeah.
We don't actually know the nonlinear functions of how they interact with the environment. That is not an empirical observable, right? And we can posit mathematical forms, and that's how we have been making progress, by making guesses as to what these interactions are like. But in ecology, there is a whole zoo of different interactions.
Mm.
They're called type 1, type 2, type 3 interactions, and then people get more complicated within them. um, and I agree that it would be very useful to come up with a methodology for the inverse problem.
Mm.
Yeah, I mean, that's inherently difficult, right? To go from a one to many, like you're effectively trying to take a coarse grained representation and make it like all atom. Or, yeah, you can't generate information, right? So it's
Yeah,
definitely
no,
tricky.
I agree. It's tricky. I don't know. Maybe there's something in the correlations. I haven't given this question a lot of thought, but it definitely would be useful if it were be if there were some way of even constraining what the underlying nonlinearities might be.
Mm. Yeah.
Very good. Shall we move on then to the next paper?
Perfect. Yeah, I
Yeah.
think it's a good
Let's do
segue
it.
as well to the next one.
Okay, so the paper, or I guess it's kind of a series of papers that I'm going to be discussing, is basically what spun out is this idea known as the Jarginsky Equality. And so, what the general idea of the paper is to say that, okay, I know if I have a thermodynamic system that, let's say I have kind of a double well potential, that the change in free energy going from one well to the other well is always going to be less than or equal to the amount of work that I have to put into the system. So this more or less just kind of drops out of the second law of thermodynamics, and that the entropy is always increasing. And whenever I move from one state to another, I'm going to be losing some of that energy or work that I put in. Some of that work that I put in is going to be dissipated out as heat. So the idea is to say that it's very difficult to compute what these free energy differences are if we just look at the work coming from the system. In order to have them equate perfectly, we need to do this quasi-statically, which if you imagine in the case of the classic example is like a piston kind of expanding or compressing down. You have to do this infinitely slowly. And anyone that's ever pumped up a bike tire does not want to spend, an infinite amount of time pumping up their tires. So when you move from one state to another, how you can calculate this is actually quite clever. You say that, okay, instead of just looking at the total amount of work that I'm going to put in and having to do it quasi-statically to keep the system in equilibrium, I guess to explain what quasi-statically means is more or less, I perturb the system a little bit, I allow it to relax back to equilibrium, I perturb it a little bit more, I allow it to relax back to equilibrium, and at every kind of step in time, I'm never really pushing the system out of equilibrium. And so kind of the beauty of the Jarginsky inequality is to say that, okay, now if I take a system and I perturb it out of equilibrium, can I say anything about the work, the amount of work that I'm putting into this to the change in free energy? And it turns out actually that you can. So if you more or less look at an ensemble of trajectories that start from one state and look at an end at another, you wind up finding that the expectation value of the exponential of the work is equal to the total exponential of the change in free energy, which is maybe not, you wouldn't exactly expect that, because if the work is always greater than or equal to the total change in free energy, the exponential term equating those for, especially for non-equilibrium kind of pulling or pushings, is quite fascinating. So I know that's kind of a lot to just launch out straight from the get-go. So do you guys have any questions about that?
Yeah, Clark, I was wondering if you could talk more about the path integral calculations that were done in order to arrive at this.
So what's actually interesting about this is there's a few different ways to derive kind of this result. So one of them involves a path integral. One of them involves like a master equation. So the path integral formulation is more or less to say that just like in quantum mechanics, I can take any path to get from point A to point B. And I'm going to weight a particular path based on some exponential function that I have. And so in this case, the exponential that we're weighting is in fact exactly just the change in the change in the Hamiltonian of our system moving from one state to another. And so taking the limit of this, as you decrease the step smaller and smaller and smaller, spits out exactly the same thing that you would get if you were to go at this straight from the get-go with a master equation.
Okay, perfect. Do you have an intuition as to why the master equation um, gives you a very similar result as the path integral? Just because, yeah.
Yeah, so I think the intuition is with a master equation, you're looking at how things kind of are moving in space, just like how you look at things with the path integral. And so it's kind of like taking a discrete versus a continuum limit of things, even though a master equation is, it can be discrete or it can be continuous. So the path integral is basically taking every single possible path that you can take, whereas the master equation is taking just kind of the average of those paths that you take. So the path integral, generally kind of speaking here, is just kind of the average. When you take only kind of the smallest time step, is you're only going to be taking what on average you would take in any typical time step. And so I guess to speak a little bit about the relevance of this paper and why I think it's kind of special is that a lot of times in biology, as you kind of hinted at before, we don't really have things in equilibrium, right? Like if something is in equilibrium, there's no flux in or out of the system, and that's not what we typically observe, right? So stationary states, sure, all the time. Things come in, things go out. But to have something where nothing is coming in or out, well, that cell's probably not going to live very long, right? Like you need new nutrients, you need new resources. And so to be able to compute what the free energy difference is between something, we can't really use equilibrium approaches very often. And so one thing that this Jarginsky inequality helped kind of rectify is if I have an RNA hairpin, right, it's this kind of Velcro-ish thing where things are glued together, you want to know what's the free energy difference from going fully kind of stuck together to fully kind of ripped apart. And so instead of having to take this thing with, some type of optical tweezer, move it out a little bit, and then wait a long time for it to re-equilibrate and do that for the entire hairpin, what you could do now instead is take this hairpin, pull it at whatever speed that you want, and you can average across all of those trajectories that you get to compute what the estimated change in free energy is. And so the Jarginsky inequality is a very nice way to basically perform experiments and also perform some calculations in kind of the experimental setups that we would actually want to be using.
Wow, thanks for the explanation. I didn't grab this thought before when I was reading the paper. For me, it was a bit cryptic, but the RNA example is fascinating. Yeah. So
Yeah,
just
I mean,
average. Yeah.
I think Jarginsky is a fantastic theoretician. And he actually kind of came up with this before they did the RNA experiment. So the RNA experiment kind of helped confirm his hypothesis. It wasn't like they had the data and then they went in and explained it with the Jarginsky inequality. In fact, it was kind of the other way around.
To me, what's fascinating with this paper is one more time you find the Boltzmann factor everywhere, and this is what rules the statistics of energy. To me, it's fascinating to find this exponential minus beta time energy as the ruling.
Yeah, I mean the Boltzmann factor is almost like the boogeyman under the bed, right? Like you can never really get away from it.
Yeah, it's underpinning everything. Like as soon as you've got some energies, it falls
Yeah, I mean, it's definitely
tiny
kind of a
factor.
fascinating approach, right? That you can't just look at a single trajectory. You have to look at an ensemble of trajectories. And anytime you have an ensemble, you're going to wind up getting a Boltzmann factor out of it.
Clark, I had a question. So, the author talks about how this is only valid for a sufficiently weak coupling. And I was wondering if you could talk more about what that weak coupling means, both thermodynamically and realistically for either biological or other chemical systems. When would this sufficiently weak coupling be realized?
Well, I don't want to plunge too far into the deep end here, but I'll say maybe like a sentence or two about what I think he means. So generally, when we talk about kind of a weak coupling, we mean we have some system and we're coupling it to a heat bath. And so if you have a weak coupling, more or less you just have something where that's not like the primary driving force for the system, right? Like if I have a separation of the force scale where the coupling dominates everything, then this Jarginsky inequality might not apply. So, kind of thermodynamically, you want to look at like what is the dominant force in the system and how it relates to the coupling that you have with either a heat bath or an external piston or something like that. You know, if you take that RNA hairpin and you yank it so fast that you're actually breaking bonds, then you're not going to get a good representation of the system.
Okay, perfect. Thank you.
So I guess we can move to Maxime now. So if you want to discuss the paper you have.
Sure. I think it's a great paper that follows the spirit of our podcast today, because it's talking about uh, population and how they equilibrate in environments, and also the importance of considering the statistics of a population to understand what rules underpin some characteristic of it. And so what I'd like to talk about today is a paper written by Alejandro Stowski, and the last author is Naama Brenner. It's published recently, like at the beginning of the year 2022. And they went a bit further in understanding the way bacteria regulate their cell cycle of division and their cell size. And to me, really the beauty of the paper is that they get this information by studying the fluctuation of several cells that divide and that they monitor. So that's the fluctuation that allows to give the information on where the constraint apply to regulate this cell size and cell cycle of division. To monitor precisely the production of offsprings of bacteria, they take benefit of the microfluidic technology, where single cells of bacteria are trapped in a channel open at one end, and they monitor only the cell which is at the bottom of the channel. So this cell is going to divide, produce two cells, and they keep monitoring the one which is at the bottom. So the raw data they got is basically the elongation of the cell with time. So what they see is like a cell elongate more or less exponentially. Then it divides, so the size of the cell at the bottom decrease abruptly, and then it increase again. So you've got like in the raw data a sort of succession of takeoff ramps, which represent the elongation of the cells with time. And because with the microfluidic you can have many channels in the field of view of microscope, you can have many time series like that for many bacteria, one for one lineage of a bacteria. So let's make a first stop here. It means that there's a different way to average a measurement done on the bacteria. You can average over time, so you monitor the bacteria at the bottom of a channel, and it produces, I don't know, for instance, a size before it's going to divide. So you follow this size, it's going just before it's dividing with time, and you can average the total size over the lineage of the cell, and this gives you an average cell size before it divides. So this is
So
one.
maybe I don't quite get the experiment here, but you have some E. coli cell, and at the top you have like some fluid that's just washing over the top. So you start growing E. coli from the very bottom of kind of a tube, and it grows up until it reaches this flow channel and then gets washed away. Is that the?
Exactly. And actually, even if at the beginning you've got two cells that got into a channel, actually, because the one at the bottom is pushing the other out of the channel, the only one that is going to stay eventually, the only lineage that is going to stay eventually in the channel you consider is the lineage coming from the cell at the bottom. And the channel are very small, so they are thin with the size of a bacteria. So basically, bacteria are aligned in the channel, pushing towards the outside of the small channel. And when it reaches the open end of this channel, as you said, a flux is going to take them out.
So the idea then is to mostly check kind of population division rates and see which one is maybe a stronger divider by using this kind of assay.
Well, what they monitor is how, for an isogenic strain like an E. coli MG1655, how for this kind of cell, the elongation rate is going to vary, for instance. So you've got many bacteria under your microscope, all at the bottom of the channel, and you can see how this elongation rate is going to vary depending on the cell you considered in the first place. So that's the fluctuation of, or the variation of the elongation rate that you can assess like this, because obviously cells have an elongation rate which is in average all the same, but it can vary depending on what they experience in the microchannel. And this is what you can quantitate with this kind of microfluidic technology. Is that more explicit?
Yeah, I think so. I mean, I'm definitely not incredibly familiar with this stuff, but it's definitely a clever idea to kind of, I think, grow these things up and kind of split them in this manner.
Yeah. So you can follow single cells and have a distribution of, for instance, the elongation rate. Actually, they do many quantifications. So on this traces, I was describing this series of takeoff ramp that you can follow in one of the channel. You can measure the size at birth, so the size of the cell just after it divided, the duration it takes for cells to divide, the exponential elongation rate of a cell when it's dividing, which is more or less an exponential growth. Then I did some more complex parameters which can be relevant to describe the growth of a single cell. It's the logarithmic fold growth of the elongation. So basically, it's the logarithm of the cell size before it divided and the cell size after it divided. And then they also characterize the fold decrease during division. They characterize also some fold change during a cell cycle and the difference between the size of the cell when it started to elongate and when it ended to elongate just before the division. So these are parameters and variables that can give you an information on how cells are regulating their cell size and the time it takes for them to divide. And I think, well, if I want to jump straight to the very interesting part of the paper to me is that they've been able, by monitoring the variability between all these parameters, to identify some of them which are very stiff, so on which the constraint apply, and some of them which are more slopey, as they say, where the constraint is pretty loose, but they have to compensate each other in order to respect the main constraint which is applied to the stiffest parameter, stiffest variable. And the way they do it is actually by studying the fluctuation, as I said, between the variability of the cell division and cell size of bacteria which are in one single channel, and the variability of those parameters between all the cells in the chip across the channel. So I think it's
So I
pretty.
guess in some ways this is just like parameter sensitivity, right? Like you're seeing which sets of coarse variables or variables that I have really make a difference for your system.
Yeah, exact. And they built upon this somehow quite usual experiment nowadays on bacteria uh, with another design of a channel to investigate what uh, the bacteria are actually experiencing in the channel, and what is going to explain most of the variability that we can observe between those variable that is characteristic of the cell cycle and cell size during the division. So what they've done is quite clever. It's actually a paper from one of the author, an experimental paper that started that. They've done a sister machine instead of a mother machine, and so the design of the channels are a bit trickier. Actually, two channels which are adjacent, which contain only one lineage of a bacteria, are linked by pair at the bottom, which normally should be just a dead end, by a small V channel. So actually, you've got uh, uh, many pairs of V channel in which you can follow a lineage of bacteria which are growing during the division of the cell at the bottom. And so like this, I think it's quite powerful because you've got the variability across the channel, which is, okay, things happen in the different channels, but you can also decipher what's happening between one of these V channel where actually the cells in those V channel must experience somehow the same microenvironment. And as before, you also have the variability between this of the cells in the lineage, which is due to because the cell behave differently with their past history or something like that. And so with this smart setup, they've been able to tell apart what variability is coming from the microenvironment that is experienced in those microchannels from the variability in the lineage. I think it's super powerful, and for me, this is a super important information because it wasn't that much thought that in such a microfluidic chamber, bacteria would experience different microenvironment in the channels. And for me, it was really striking to learn that actually they experience a very important microenvironment change in those channels which are close to each other actually. And this change is going to explain the variability you have in your data eventually. So it turns out that the question is how can you investigate the statistic of a variable in monoclonal strain of E. coli with a tool that gives you precise measurement with reproducible measurement for every cells. And it turns out that this is really hard. And to find out if variabilities is coming from the way you measure things or from the actual biology which happened behind that, is you need to think hard to have a good experiment that make a good control to assess this kind of question.
Very interesting stuff. Yeah, I mean, it's definitely hard to disentangle the effects of what's noise and what's actually the signal.
Yeah, yeah. But and I think what is really cool with this paper is they show a way to learn things from the noise. So in the end, they say, okay, we've got all those variables which regulate or represent something in the cell cycle and the division of the cells. And they said all these variables are constrained, but some of them are stiff and some of them are sloppy. And actually, the stiff variables are acting on the sloppy one, such as they compensate each other in a manifold. So they show a manifold eventually where actually, so all the variables that are accessible to quantify your experiment and the one that experimental is usually are constrained by something, and there's a hierarchy between all those variables. And I think it's important to consider then, okay, there's a constraint. It applies on some variables. Maybe you never looked at the good variable to assess the question you were asking for the cell. And you need to be very careful in choosing the stiff parameter to describe what you want to learn about the cell. Because the other one, which are sloppy, are going to compensate each other.
I see. So you want to be studying around the pocket of whatever metastable state you want to be looking at. And if you perturb it too much with a, um, what did you say, sloppy and tight variable? Are those
Sloppy and stiff, yeah.
stiff?
You pick
So
those
if you
one.
perturb a stiff variable, you might not be in the regime that you want to be in.
Yeah, 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 yeah.
All right. Well, I don't have any other questions. Avanish, do you?
No, I mean, I think you guys touched on a lot of things related to what I was primarily kind of blown away with this result was showing how there's qualitatively different correlations on short time scales and long time scales, and that for me it's very counterintuitive. And I think a lot of people, when I've seen data shown where people assume a certain correlation on a short time scale but then show it on the long time scale and vice versa. um, I found this to be a great mathematical explanation of why these correlations can disappear from short to long time scales.
Yeah, yeah, that's true. Then they also show that this correlation is quite reproducible across experiment from different data sets that have been measured by different team in the world. So, I mean, the way they tackle the problem is super robust to me. I'm very impressed by that.
All right. Well, in that case, I guess we can go ahead and kind of wrap up. So I will link all the papers in the show notes. And for everyone tuning in, I hope you'll be eagerly looking forward to more Avanish and Maxime as they, help add in more episodes. So anything else you guys want to add?
Great pleasure to be part of it, and let's do more podcasts.
Yeah, absolutely. It was a great pleasure talking to both of you, and hope to also get some questions from the listeners, because I'm sure that many of us, like, we are not necessarily completely acquainted with all of the systems that we were describing, and there's a lot of extensions, and we'll be posting links to this in our Twitter channel. So if people want to engage in a discussion there, we are open to learning about what we got wrong, what we didn't understand, and what more there is to explore. So, looking forward to a great discussion online as well.
I'm sure there's a lot we got wrong and forgot to include. So, All right. Well, thanks for tuning in.